Thank you everybody for coming and attending the presentation here today. My name is John Iannone. I'm one of the program managers at Toxikon, and we're going to be talking about biocompatibility, and I'm going to do my best to give you a simple understanding to what can be considered by many as a very complex topic. So for this presentation, just a quick overview, tell you a little bit about Toxikon, where I'm coming from. Then from there, we're going to talk about what is biocompatibility and why do we even need to care about it. From there, we'll talk about some of the regulations and guidelines that are pertaining to biocompatibility, and talk about what biocompatibility testing do I need as either a medical device manufacturer or maybe even a supplier. From there, you're going to want to talk about what biocompatibility testing you need and understand what those tests are. So we'll explain what those tests are, the different categories, and we'll conclude the talk with a few lessons learned, things to remember from this presentation, other than, the more important things that we talked about before we started. So quickly, Toxicon, quick overview. We are a contract testing lab, headquartered just outside of Boston. We do have laboratories there at headquarters and also in Leuven, Belgium. We have over 35 years of experience in safety testing for the medical device and pharmaceutical industry. That includes biocompatibility testing, material characterization such as extractable leachable testing, even toxicology assessments. We are FDA registered and 17025 accredited, do testing both to GLP and GMP compliance levels, and we have over 150 researchers. Just to give you an understanding of how the organization is broken down, we have four main groups within the organization. Microbiology, doing a lot of your lot release testing as you're sending out your product, maybe the reusable device cleaning validations that you may have heard about recently, as well as the antimicrobial efficacy testing if you're getting into that realm for product development. We also do the biocompatibility testing, which is going to be the focus of our talk here today. Complementing that is both the toxicology, either in devices, combination products, or pharmaceuticals, as well as the analytical chemistry, that extractable leachables I was telling you about earlier, and even some of the bioanalytical support for the toxicology testing. So let's get into our talk and get into biocompatibility explained. So what exactly is biocompatibility? Well, in its simplest sense, it's a material's property being biologically compatible, either by not eliciting a local or systemic response in a living system or tissue, okay? What that really means is if I present a material or a device to the body doesn't really care that it's there, okay? It's very material focused. Not to be confused with efficacy testing, where you may have a certain response that you're looking to have. Biocompatibility here, where our response that we want is no response, right? From a regulatory standpoint, it's really a series of tests that help you to identify and determine whether or not a material has the potential to cause an adverse reaction, either by the components, materials, or the devices itself when they're in contact with the body. So what we have here is either the leachates of the device, okay, or the device itself should not produce any adverse local, systemic, tumorigenic, reproductive, developmental, et cetera, effects. So who really needs to care about biocompatibility testing? Who has to do it? Well, any medical device manufacturer is going to be ultimately concerned with biocompatibility. When they're going to do their regulatory submission, getting a product out to market, they need to demonstrate that that product is going to be safe on the market. And they do that through a few different vehicles. Like the 510K process or a pre-market approval. Again, anyone that is producing a final finished sterilized device cares about biocompatibility. Now we have a few people in the audience that are also raw material suppliers and component manufacturers. These individuals don't need to do biocompatibility. However, the device manufacturers often require proof of biocompatibility. When they want to bring in a material and they want to use it for their device, they don't want to be starting from ground zero and find out that they did the wrong selection of a material, so they want to see some degree of biocompatibility even on the raw materials or components. And that goes into the conundrum of, well, polypropylene is polypropylene, which is polypropylene, right? Well, not exactly, and we'll talk about that. There's a variety of different factors on how the raw material suppliers make that polypropylene or whatever material it is, or that component, and their quality systems that they incorporate into their organization can control whether or not the polypropylene they put out on a Wednesday is the same one that they put out on a Friday. In addition to that, the proprietary, right, manufacturing processes that they've come up with that makes them unique to their competitors could be also incorporating other potential contaminants into that process, different manufacturing residuals, maybe some old release agents, things of that sort. Then we also have certain polymer additives. These additives are purposely put into the formulation when you're making your raw material, so you get certain properties: rigidity, flexibility, clarity of the polymer. Maybe you want to be able to protect it against degradation or photostability. So these additives are purposely put there, but in some cases, either the additives or the resulting products from those additives could affect biocompatibility. And then lastly, a lot of things people tend to forget is even the packaging that you put your polypropylene or whatnot into could have an effect on the overall biocompatibility of that raw material, those components, and the final device. So we're going to talk more about how we deal with all of that. And over the years, in order to address biocompatibility, there really has been an evolution to the testing matrices that guide us in how to conduct biocompatibility testing. And that started all the way back with the USP. I'm sure everyone in this room, whether you know about biocompatibility or not, has heard of Class 6. Okay, Class 6 has really become the foundation of being able to say that a material is medical grade. Okay, there's a series of tests, a few different extracts, and at the end of the day, we say it's Class 6 approved or not. And that's really the start of biocompatibility, following the USP Monograph 88, looking at biological reactivity of plastics. Well, with that, devices are made with more than just plastics. So what happened was in 1987, the FDA came out with their tripartite biocompatibility guidance document. This is what formalized how to think about biocompatibility, how to get through biocompatibility, and different devices, how to assess that biocompatibility that's pertinent to you. From there, it was very evident that there needed to be a international understanding of what biocompatibility is. As the world got smaller and more people are outsourcing overseas, we wanted to all understand biocompatibility. So the ISO 10993 took it upon themselves to pull in experts from around the world to assess and determine what is the thoughts and considerations that we need to have in order to determine if a product is biocompatible. With that, the FDA said, hey, this is a great document, let's talk about it some more. I'm going to tell you how I want you to read that document and how I want you to interpret it and what will make me happy as the FDA when you go to follow that ISO 10993, and maybe think of a few things that I thought of. So that really happened initially in 1995. And most recently, back in April of this year, they came out with a new draft guidance, which is going to be going through final comments, I think the beginning part of next month. And that is, again, how to use the ISO 10993, particularly Part 1, where you find out what tests you need to do for your product. And we'll talk more about that in a little bit. The Japanese also said, hey, ISO 10993 is pretty good. I got a couple ideas myself. So, we were all trying to conform to what the whole world can look at, but a couple guys said, okay, well, here's what I think is missing, or let's do this. And for the most part, through that evolution, everyone really has come together, and even ISO has been considering the comments from other regulatory agencies and have come together with a nice understanding of what biocompatibility is, regardless of what the individual companies are looking for or individual regulatory agencies. And with that, you can pretty much say that the majority of testing is really performed to the ISO standards. So what are the ISO standards, ISO 10993 standards for biocompatibility? There's about 20 parts, and they're all under the general title for looking at the biological evaluation of medical devices. What I have here is parts 1 through 12, and we're going to focus mostly on parts 1 through 12 that I've highlighted here in red. Okay, these are going to be how to determine what biocompatibility testing is required, how to conduct some of those biocompatibility tests, and either the in vitro or in vivo aspects. The 10993 series does go into 20 total parts. Parts 13 through 20 are predominantly focused on material characterization, the chemistry, degradation, and how to assess that data, which will be presented on another talk. So what does that testing matrix look like? Looks just like this. What we have is a variety of different mechanisms that need to be assessed for biocompatibility, depending on the concerning levels on how the product is exposed to the patient. And which test to select, with all these little squares and dots on the screen, is determined by how your product is in contact with the patient. You know, what's the body contact, what's the duration of contact, and here we talk about how to correctly categorize your device. Is it a surface contacting device? Is it external communicating? Maybe it's an implantable. And under each one of those three categories, they're further subdivided for either skin contact, mucosal membrane contact. We even have breached or compromised surfaces, all for skin contacting. And those are treated a little differently here and there for the biocompatibility. For external communicating device, you could have indirect blood contact, contact with tissue bone, or maybe indirect with circulating blood. And for implantable devices, either the tissue bone or directly with blood. So the next part I talked about not only the type of patient contact, but maybe the duration of contact. And the way the devices have been categorized is it'll either have a short-term exposure or a limited exposure, as it's listed in ISO 10993, prolonged exposure, which goes greater than 24 hours but less than 30 days, and then permanent exposure, which I want to identify as greater than 30 days, not for the rest of your life. It's defined from a regulatory perspective, permanent exposure, greater than 30 days. And what I have here is a variety of different examples, right? And we have our, catheter that may have some prolonged exposure. We even have some surgical instruments that could be for a limited exposure, and some permanent exposure implants. Obviously, all of these devices need a different level of testing, and that's what the 10993 matrix that I was showing you earlier helps you to discern. One of the things that isn't very well understood in the industry is considering this repeat exposure accumulation. So here I have the example of a meter dose inhaler. Well, that meter dose inhaler is used for a couple seconds, right? Well, that meter dose inhaler is used for a couple seconds over and over and over again. So there are further guidances that talk about certain devices like the meter dose inhaler and how to address it more like a permanent implantable device. For sample preparation, so we've got an understanding of all these different tests we have to do. We're categorizing our device. We're saying, okay, I'm going to get there. I'm going to be doing my biocompatibility testing. Well, how do I do it? My device is this big. What are you going to do with that? Well, sample preparation is actually pretty well defined, and you want to focus on using final finished devices. Everything you do, like we talked about earlier, those manufacturing processes, that packaging, sterilization, these can all impact the chemical profile of that material and therefore how that may react with the biocompatibility tests. Most tests in biocompatibility are actually conducted with extracts of that device. So that's how we get the big devices to be able to go into the different small test systems. And one thing to note is after extraction, these devices really aren't suitable for use. When we do those extractions, we're trying to extract what could be exposed to the patient. So the chemical profile changes, what's left over changes. You may have lost some of those additives that were helping to maintain the flexibility. So the mechanical strength may be affected. Certainly the biocompatibility is now affected, and a variety of different things. But they're really great for being able to do show and tell. And extractions are always going to be based on a certain surface area to volume ratio or weight to volume ratio. So it's very well defined. In Part 12, they say things like 6 centimeters squared per ml, 3 centimeters squared per ml. We're not going to get into why they chose that, but they're very well defined, and the FDA and other regulatory bodies really like to see that you maintain those extraction ratios for your testing, is to understand now, we're talking about biocompatibility of devices. The other thing that we test in the industry for safety is drugs, right? Well, with drugs, we're looking at different dose levels. We're going to look at high doses, medium, low dose, therapeutic windows. And depending on what we get out of our tox data, we may adjust what our dose is going to be. Well, there's very little room for interpretation in biocompatibility testing. Those studies have been well set up so that you can test your catheter system, your Band-Aid, your stethoscope, whatever is appropriate. Things that get a little more obvious is things like hemodialysis catheters or pacemakers. If you go to test that pacemaker and with that really tight pass-fail criteria you did not pass, you can't really say, okay, well that was my high dose, I'm going to use 10% of a pacemaker and then I'll just use that for my patient and then it'll be safe. Well, 10% of a pacemaker, I promise you, because my background is in engineering, it won't work. So you really need that whole pacemaker. And really that's the notion behind why those surface area requirements are there, why those exposures are there. They're thinking about the sensitivity of the different tests in biocompatibility as well. So for the test selection, again, biocompatibility tests are very well defined. It's really important to consider what is adequate by the agency and resist the temptation to try to reinvent that test. You know, they're very well accustomed to the tests. They're very well laid out in the 10993 and the technical documents. So you really want to avoid the, okay, well, I think if I do this just a little differently, it's going to be better for what my device is used. As I said, the FDA is really accustomed to certain test structures, and you want to stick to what they have seen historically. And really, if you've identified a deficiency, you know, again, my background's in biomedical engineering, we always find why the tests don't really assess my product the right way, the way I'm going to use it for my patient. If you've identified some deficiencies, there even is some language in the 10993 that says if your product is not properly assessed for its safety, not efficacy, then go back and see what additional testing you can do. You know, what supplemental testing. Try to keep the tests that you're doing that are well defined intact by themselves. Don't hack them up and say, okay, well, what else can I do to really show that this is safe because this wasn't well captured? So that's an important aspect. Now, getting into the cytotoxicity testing. We don't use birds for cytotoxicity testing. It's an in vitro assay. It's cell-based. But really, what it's come to be known in the industry as the canary in the coal mine, right? It's so you know that when you're starting your biocompatibility testing, you have a fair assessment of whether or not that product is relatively safe. Now, granted, we're looking at one particular mechanism, but it's a sensitive mechanism, and it helps me understand the overall biocompatibility of my device in its simplest form. And that's the theme of this talk, right? Biocompatibility, simple. There's a variety of different assays, agar diffusion, MEM studies, MEM elution, the NRU, and they all have their pros and cons. The selection has a lot to do with what you're trying to mimic for your device, what the device is compatible with in terms of different assays. So really, there's a couple options available to you. And we really won't get into what each one of those are, but certainly if you want to talk about it, I'll be available after the presentation. So irritation testing. Nobody wants anything that when you put it on your skin and you're going to get a rash, right? So we have to test to make sure that you're not getting any kind of irritation, whether it be dermal, or let's think about our implantable devices or external communicating devices. You don't want that irritation happening inside. So we could also be looking at intracutaneous reactivity, which is just a fancy way of doing irritation in a particular area inside the body. There's also the understanding that you need to understand your device's site-specific requirements for irritation. If something is not going to be an irritant on the skin, well, we all know that same thing, if I got it in my eye, it's going to hurt all heck, right? So we want to look at the irritation for ocular irritation, or maybe mucosal irritations, and a variety of different other site-specific irritations depending on how your product's used. Another mechanism for testing biocompatibility is sensitization. So here, we're looking for whether or not that material is going to induce some kind of an allergic response. First time you touch poison ivy, nothing happens. You say, great, didn't matter to me. I can go in my backyard and roll around in my boxers, I'll be fine. But if you do that 2-3 more times, next thing you know, you say, wow, I need to load up on a lot of hydrocortisone and deal with this now. That's sensitization. So you're building up antibodies in your body that say this chemical and you don't really go together. Whether or not your material can induce that response needs to be assessed. And that's the sensitization testing. A few different assays to be able to do that. Here we have listed the guinea pig maximization test, also more commonly known in the industry as the Kligman. Okay? The closed patch test, that's actually the Buehler adjunct patch test. And then there's also the mouse local lymph node assay. Few differences on how those tests are conducted, a few pros and cons on what that all means. One of the things I'll point out is that we have the challenge to induce that allergic response in a short amount of time. So we want to be able to do that test and get that response. And so with the Kligman, you actually use an adjuvant to help induce that response and a repeat exposure, and then do the assessment at the end to see whether or not you saw any kind of evidence of sensitization. The Buehler patch test doesn't have an adjuvant, and the mouse local lymph node assay kind of takes care of that to a good degree, but it's not very well accepted, or I shouldn't say it's not fully accepted, and I'll say that yet. So stay tuned on that one. So moving down in the biocompatibility testing, we've got acute systemic toxicity, pyrogenicity. That's really when we have anything that is going to enter the body. And we want to know, okay, well, how is that going to affect the body overall? There's going to be some kind of systemic effect. So for the testing, an acute systemic injection study, we have a very high dose of that extract. That extract is a high concentration of stuff that comes out of your device. And we're going to look for over 72 hours whether or not we get any kind of clinical observations. If the mice don't like it, they're going to show us, right, for that acute response. Another part of that is actually pyrogenicity testing. And pyrogenicity testing, for a lot of you that know your microbiology, you can be looking at endotoxins. That's all the microorganism stuff, and we can talk about that on another talk. But what that can do is it can actually cause the body to spike a fever, okay, cause that fever response. And that's not really good either when we're dealing with critically ill patients, in the ICU. We want to make sure that that's not an issue. So how do we do that? We can be looking for endotoxins from the microorganism standpoint, but there are also chemicals that can cause that same response that you won't be able to capture in the endotoxin testing. And the only way to do that is really to dose that to an animal and actually look to see whether or not there's a rise in temperature. Continuing with the systemic toxicity testing, we talked about acute exposures. Well, what about the people that have a device that's used repeatedly? We talked about our inhalers. There are other devices with that repeat or continuous exposure. Now we want to look at subacute, which is a misnomer, or subchronic systemic toxicity. And here we have that repeat and continuous use that's being exposed to the animals. And then we do a variety of different assessments, because this is usually for a higher concern application, material, delivery to the patient. We're going to do histopathology, looking at different tissues, organs, not only at the implant site if we were to implant it or IV inject the extract, but critical organs. Are they all behaving appropriately? Is there some kind of reaction happening there? Different kinds of clinical chemistries. Is my blood, clinical pathology, is my blood now starting to react in a certain way and saying, hey, I don't like what's going on, what are you doing to me? Looking at organ weights, are the organ weights changing? That could be an indication. And a variety of different things. We're looking then at a statistical comparison of how your test article compared versus a control article, okay? And whether or not you had any worse of a reaction, or hopefully no reaction, than you did from the control. One thing I want to note is these tests, you may hear them as like 14-day repeat dose or 28-day. Well, they take a lot longer than 14 or 28 days. They can be, longer in duration. They can be costly, so you want to plan ahead for that. And genotoxicity testing. Here, we're really looking and screening, okay, for the potential that your material or its leachates can induce the potential for cancer development, right? That genotoxicity. And it's not to be confused with reprotox. Even though in the ISO 10993 they kind of group them together, there's some relations, but what we're focusing on right now is genotoxicity. And there's a few different mechanisms that can cause genotoxicity, like DNA damage, gene mutations, chromosomal aberrations, a lot of stuff that the scientists eat up all day. And what we want to do is make sure that our product, depending on what the concerning level of exposure to the patient is appropriately going to be assessed for these various different mechanisms of gene tox as appropriate. A lot of people know the different tests like the Ames assay. If you're doing gene tox and you don't have really a long exposure or permanent exposure or an implant, chances are that's the only assay you need to do. But in many cases, you need to do other tests like the chromosomal aberration or the mouse micronucleus assay, perhaps even the mouse lymphoma as listed down here. And then there's implantation testing. So if you're going to take a material and implant it into a human, and we want to make sure that, as we said before in the definition of biocompatibility, you want to make sure that the body doesn't recognize that it's there. Well, everything we've been doing is helping to assess that, but this is probably one of the best tests to make sure that the body doesn't recognize that something's there. Put it right there in the body. See what happens. Is there going to be any kind of tissue, any kind of infiltration? Is there going to be, an immunogenic response? Is there going to be some kind of growth there? And the pathologist can review those slides and actually start to say, okay, compared to my safe predicate, this device is starting to have these kind of reactions. And they can score it and determine how bad of a reaction it really did or didn't have. All the different sites and tissues, like we talked about irritation, they can also have a different response from implantation. So you have to consider your device's use and what's the appropriate site for implantation. What that doesn't mean is that just because your product is a drug-eluting stent, that you have to go into the artery, for example, or whatnot. There are other areas like intramuscular tissue that's highly vascularized, and this goes back to doing something that the FDA is used to seeing. They're used to these types of assays and these types of implantation sites. So be careful about how much you're going beyond the requirements and maybe going into an area where an FDA reviewer might say, okay, I've never seen this before, now I'm getting a little nervous, okay? Another really important thing here, we're comparing to contralateral controls within that same animal, and what their reactions are. Well, in that case, a predicate device can be leaps and bounds useful in being able to determine whether or not your device is really causing a reaction or the predicate that has been determined as safe is causing no worse than a reaction than yours. It's a very important concept, especially when you get into certain materials that are like mesh-like. You can get different types of tissue ingrowth that could be misrepresented as toxicity, but if you have the right predicate, you can deal with that more appropriately. And how long your device is implanted is also a really important part. So we talked about an implant. A lot of these tests, we were just going to the end of the study. Longer and longer that your implant study has to occur, the more you're going to want to assess at discrete time points. Because let's face it, if we're doing an implant and that implant is for 26 weeks, we have no idea what happened at 4 weeks with that tissue, because we're not going to cut a piece of the tissue out while the animal's still there. So we want to have multiple time points to be able to look at what kind of response happened and whether or not the animal recovered from that response appropriately. Did that response at 4 weeks really present an issue that even though at the end of the 26 weeks we thought we were good, that 4 weeks was not so good? And hemocompatibility. So we talked a few times about devices that are in contact with circulating blood. Well, there's a lot of things that you can do to assess whether or not a product can be in contact with that circulating blood, either for a short period of time or a longer period of time. For hematology, the hemolysis assay is probably the most common assay that you would want to assess for contact with blood, either direct or indirect. But there are other different mechanisms to evaluate whether or not you have hemocompatibility. Are you actually causing coagulation to happen faster or slower? Do you induce a complement activation? which is a little bit of looking at immunogenicities and also looking at whether or not you are hindering the clotting cascade itself. Also platelets. Platelets have to do their thing. Is your device making them all aggregate to the device? You know, that's not really going to be what you want happening in circulating blood. Also thrombosis, thrombogenicity. That's another important aspect if you have a device directly in maybe a vein. And finally, all this hemocompatibility testing to varying degrees is required for any device that contacts blood, either for a short period of time, longer periods of time, or circulating blood. So with all that, we have a few closing comments. It's important to test your final finished device. That's really what you need to test for your biocompatibility. As we said, a lot of the different processes, additives, they all can affect biocompatibility. So don't use a prototype that had a different material or a different manufacturing process because you may not have captured something appropriately. Don't wait until the last minute to initiate your testing. Some of these tests need some careful planning, whether it be for sample requirements or just planning your budgets, you know, planning when you're going to have things done in time for your regulatory submission that you set up. Don't ever set up your regulatory submission date without understanding what you're going to do to get there. Don't test too far in advance. Now, I just said don't wait too long, but also you have to balance it. If you go too far in advance, there are things that can happen. Guidelines change, you know, how you do something can change. The regulatory body you're going to be submitting to may have a couple changes. Devices actually change, you know, design freezes. If you haven't gotten to a design freeze, be careful about jumping into your full biocompatibility. Certainly, some biocompatibility and that class 6 testing I mentioned earlier, very useful in material selection, evaluating as you're going through your product development process, whether or not you're incorporating or inducing anything that could have an adverse reaction. Very helpful there, but the full range of biocompatibility testing, you know, again, wait till you've gotten to your design freeze. You know that this is your device moving forward and your manufacturing process. Also, the guidelines are always evolving and changing. As I said, the FDA just released their guidance to the industry on how to interpret the ISO 10993. That happened just under 2 months, just over 2 months ago. So understanding what the regulatory bodies are thinking in terms of how they want you to do that testing is also very critical. Fully understand the ISO 10993. It's great bedside reading. Okay, so look at parts 1 through 12. And I know I didn't talk about any of the chemical characterization, but I promise you, if you also read Part 18, you're going to find some additional value there. So Parts 1 through 12 and Part 18, start there, understand that before you dive into biocompatibility testing. Don't take any paragraph out of context. You know, there really is an assortment of all these different guidance documents that are interrelating to each other, different draft guidances that refer back to the ISO documents, and you want to understand biocompatibility as that regulatory body looks at it as a whole, okay? And then finally, I'm going to say it's really important to talk to a lab that really specializes in ISO 10993 testing and biocompatibility testing, just so you can get a better understanding of what am I getting myself into? Here's my device, this is what I'm doing. What tests do I need to be considering? You know, have that initial discussion. I'm friends with a lot of people at a lot of different contract labs, and we all have that same mentality. We're willing to have that conversation, you know, hour conversation, and then you can go on for the next 6 months or whatever, doing your thing, and at the end of that 6 months or a year, you're now more prepared because you knew that your biocompatibility is going to take 2 months or maybe 9 months or a year, I don't know, whatever you have. So having that conversation early on can really be very worth it. And what I'll say is have that conversation with various different groups, not just with the product development or regulatory, but really incorporate other aspects to your organization. Marketing, because marketing really wants to put on their label that the product is going to come in blue, red, and green, but then after you start talking about things, you may find that that has its own issues in and of itself. So with all that, I want to thank you for your time. I hope this was a little informative about biocompatibility. And I think we're out of time, so if anyone wants to ask me any questions, I'll be over at the Toxikon booth 2900. Thank you.